Hello, everyone, and welcome to another. Hello, everyone, and welcome to another episode of Burda Biology. For my last lecture in cells, I'm going to be talking about two main things. So let's kind of try to figure out what the takeaway is from the last bit of cells. So we've already talked about compartmentalization in cells, the differences between prokaryotic and eukaryotic. Second lecture, I talked about the main membrane-bound organelles found in eukaryotic cells. From my last lecture in cells, I'm going to be talking about the organelles involved with information processing, so the conversion and reading of DNA and genetic material, and also organelles involved with energy processing, which we've already talked somewhat about those as well. So let's go ahead and get into the information processing organelles. And this is going to be the one that is found in both prokaryotic and eukaryotic cells. And that is involved with the ribosome, which we finally get to talk about. But first, let's talk about general information flow. So here we have, it looks like there's a lot going on, but I'm going to help decipher this for you guys. So we start in biology. This is called the central dogma of biology. It's kind of the essence of what biology is. So how do we go from DNA to us? It's kind of an abstract idea, just to think that all of our code for our blueprint to create us is in that little strand of DNA. But what's going to happen is you go from DNA inside of the cell nucleus. We go through a process of transcription, so it has to be read, and we're just going to copy into a small little sliver because the whole DNA, the whole piece of DNA cannot fit out into the cell. So let's just make a copy or a code for one single protein. That's when we transcribe it into a molecule called RNA. RNA can then be translated, like a textbook in a different language. It can be translated into a protein. So every 3 letters in an RNA molecule is going to code for an amino acid. Each of these letters here is representative of an amino acid. So that's how we go from our blueprint actually to what we look like. From DNA, DNA goes to an RNA, RNA goes to a protein, transcription and translation. So it's a pretty, gets pretty in-depth, but we're just keeping it minimalistic for now. So these guys are essential in the conversion of the RNA into the protein. So these guys are involved with the translation process. This is the ribosome. The ribosome is found in both prokaryotic and eukaryotic cells because, after all, prokaryotic cells need proteins as well. They're made up of 2 main subunits, the large and the small subunit. They can exist in 2 locations within the cell. They can be floating around in the cell, and we call those free ribosomes. They're just floating around there in the cytosol. Or they can be bound, and those are the ones that you found stuck on the edge of the rough endoplasmic reticulum. So they can be either free or bound. And ribosomes, like I said earlier, are going to actually convert that RNA strand. So here you see the RNA. So this is just one section of the DNA coded into an RNA strand. And this thing will equal, when it's all said and done, this will code for one single protein. So this actually gets run through the ribosome in between the large and the small subunit of the ribosome. And it's going to turn it into, right here, a chain of amino acids, which you call a polypeptide. You should remember that from your macromolecules unit. And out comes this fresh, newly formed polypeptide that's ready to be turned into or folded into a mature protein. I'm not going to get into this whole process. You see a lot going on here with tRNAs and all that. That's for later uses. I just want you to understand that it's converting nucleic acid, ribonucleic acid, into a polypeptide chain. So going from an RNA, translating it into a baby protein. The endomembrane system, again, is involved with that flow of information in the cell. Remember, right now we're talking about how does information flow within the cell. So the flow of information, I talked about this in my last lecture. You're going from the nucleus, going from DNA to RNA. That goes out of a nuclear pore into the rough ER or the smooth ER, where we can make lipids or proteins. That then gets transported in a vesicle to the Golgi. The Golgi packages it, modifies it, and then ships it out in a vesicle where it can then be brought out to another cell or used within the cell. And a little side note here, cytoplasmic proteins are made by free ribosomes and may not move into the ER. So they don't necessarily have to go through this process. You can have some little ribosomes just floating around out here, just simply making proteins all the time. So it doesn't necessarily have to go through that process. And for the last bit, the last little section here, I'm going to be talking about are the organelles involved with matter and energy processing. So which are the ones that kind of transform or convert different forms of energy within the cell for essential processes. And this is almost just a review of what we talked about before, but just so you're clear, these are very important. This is the mitochondria, just kind of a cross-section of it. The inner membrane, we actually call the cristae. It's kind of an odd name, but here would be the cristae, the inner membrane. And it contains many copies of the enzymes needed to produce ATP with maximized surface area. So the enzymes that we would be talking about, that's where we're actually going to have ATP synthase. But you know, when we talk about energy processing later on and how we actually make ATP, this is going to make a lot more sense to you. But notice we have so we have this membrane here, we have the cristae, and then there's kind of like all this space in between here, and that's going to be essential to the the structure aiding the function of the mitochondria when we learn about that later. So that's going to maximize the production of ATP. It's quite a slick little machine actually. And then the chloroplast is kind of similar to the mitochondria here, but it actually uses sunlight to convert it into another form of chemical energy, sugar. So the chloroplast contains thylakoids. Sorry. A double membrane with an inner membranous stack called a thylakoid. The thylakoid membrane contains many copies of the enzymes and chlorophyll needed to produce chemical energy from solar radiation. So chlorophyll is actually the pigment used to absorb that sunlight. So it can be then converted into sugar. The stroma is going to contain the enzymes needed to produce organic compounds. So this would be the stroma. It's all this fluid-filled space out here. Here's the label for it. There's the stroma, and it says it has all the organic compounds. So those would be your carbon-based things like CO2. After all, sugar is C6H12O6. So plants get their organic compounds from the air. So they convert CO2, they fix the carbon, they use that, and with the help of sunlight, then they can turn that into sugar. So they use the energy from the sun with CO2 to actually fix the carbon and then turn it into sugar, which don't get yourself all wrapped up in that. That's what we start talking about later. You just need to know the basic structure and function of the chloroplast. And this is unique, and this goes back to the idea of the endosymbiotic theory. But mitochondria and chloroplast both have their own DNA, which is quite interesting. And it's circular. And as you know, our cells have DNA that is in the double helix. So that's quite different from the circular DNA in a mitochondria. So how is it that our cells differ so much from mitochondria? Well, that's because we think at one point mitochondria was its own separate living organism. And same with the chloroplast, same premise. So that goes back to the idea of the endosymbiotic cell theory. The last little thing I wanted to talk about before I wrap everything up are lysosomes. And lysosomes are pretty cool. They're actually collections of digestive, really strong digestive enzymes. We actually call them hydrolytic. Hydro you may think of as water, lytic you may think of lysing or breaking things apart. That's how they got their name Lysol. It destroys or kills germs. So they digest molecules, they can recycle cell parts, break them down into their smallest parts, and sometimes cells need to commit suicide in a process called apoptosis. So apoptosis is programmed cellular death. If a cell gets infected with a virus or it's just too old and creaky and clunky to do its job, the cell, for the good of the order, will actually kill itself. And it does that with the help of a lysosome. The lysosome will inject its deadly hydrolytic enzymes, thus dissolving it. So it's used in several processes: digestion, removal of waste, and programmed cell death. And here I just have a little bit about vacuoles. Vacuoles, these are the storage containers, the gladware, if you will, of the cell. They can be used to store waste to maybe later be removed. Store in plants could be pigments, water, things like that. Plants have this huge one though, called the central vacuole. The membrane surrounding it is called the tonoplast, if you were wondering what that is. So the whole thing is called a central vacuole. The membrane surrounding it is called the tonoplast. And then animal cells do not have that massive vacuole, the central vacuole, but they do have little tiny ones used for storage. So that wraps up all three of my cell mini lectures. And this final cell lecture, I was talking about the transfer of information flow in a cell, going from DNA to RNA to a protein, and how it moves around. And then finally, I wrapped up with how energy is transferred and the main organelles, their structure and function, how that's all related to the transfer of energy. So hopefully you can tie all three of those lectures together, and this was a useful thing for you. If you have any questions or comments or anything you would like to add that might be helpful for you in these videos, please post that in the comment below. And thanks for listening in. I will see you in class.